This is Group 4's Lab Report 10 on PCR by Lucas Perez and Vianka Sanchez. Introduction: Food safety and the avoidance of diseases brought on by widespread bacterium depends on the detection of E. coli in food. This lab looks for the E. coli gene in food samples using the polymerase chain reaction, or PCR, a method that amplifies particular DNA sequences. This experiment emphasizes the significance of recognizing pathogens in food to reduce potential health hazards and maintain public safety standards by creating primers to target E. coli DNA, as well as evaluating the findings using gel electrophoresis. The materials needed for this lab include the PCR thermometer, the PCR tubes, which had a 25 microliter capacity, food samples suspected to contain E. coli, PCR masseter mix, which contains the Taq DNA polymerase, the DNTPs, the buffer, and the MgCl2. We also needed a forward and reverse primer specific to E. coli gene, nuclease-free water, positive control DNA, which was our known E. coli DNA, and the negative control DNA, which was the no DNA template, electrophoresis equipment and gel, the DNA ladder marker, the gel loading dye, and then the Argos gel. Methods. The first step was the PCR setup. So first we labeled the PCR tubes according to the samples that were going to be tested, which were food sample 1, food sample 2, positive control, and negative control. We then prepared the PCR masseter mix and added 8 mLs of the forward primer and 24 of the reverse primer to each PCR tube. We added 1 microliter of the DNA template, which was extracted from food samples, to the corresponding PCR tubes. We then added 22 microliters of the PCR masseter mix to each PCR tube, and we vortexed the PCR tubes gently to ensure proper mixing. The second step was the PCR amplification. So first we placed the PCR tubes into the PCR thermocycler and we ran the following PCR program. So the initial denaturation was 94 degrees Celsius for 3 minutes, denaturation 94 degrees Celsius for 30 seconds, annealing was 57 degrees Celsius for 30 seconds, with the extension being 72 degrees Celsius for 30 seconds. The number of cycles were 35, and the final extension was 72 degrees Celsius for 60 seconds. Continuing with methods, we have step 3, which was the gel electrophoresis. So we prepared a 1.5% Argos gel using 0.7512 grams of the Argos, which was dissolved in TAE buffer. We then added the gel red RM or other DNA stain to the gel, and then we loaded the PCR products, where we used 20 microliters into the wells of the Argos gel along with the DNA ladder. For that one, we used 10 microliters, marker for size reference. And then we ran the gel at 150 volts for about 30 minutes until the bands were sufficiently separated. And then for step 4, which was visualization and analysis, we visualized the gel using a gel documentation system under the UV light. We then analyzed the PCR results to determine the presence or absence of the E. coli gene in the food samples, and then we compared the results to the positive and negative controls. The last 2 steps for methods were 5 and 6, 5 being the interpretation. So if the PCR product shows bands of the expected size, then it indicates the presence of an E. coli gene in the food sample, suggesting a potential food safety concern. And then if the PCR product shows no bands, then it indicates the absence of the E. coli gene in the food samples, which is reassuring for food safety. Lastly, 6 was the food safety assessment. So based on the PCR results, we had to assess the food safety risks associated with the presence of E. coli in the food samples, and then take appropriate measures to ensure the safety of the food and prevent potential foodborne disease. Results. So starting from left to right, we have the very first one on the left is our negative control, and that faint band in that lane is likely due to the use of a TAE buffer instead of nuclease-free water in the gel preparation or running buffer. The TAE contains trace amounts of nucleic acids or impurities that can sometimes produce non-specific fluorescence under UV light, and it doesn't indicate that there's contamination in the PCR itself, but rather an artifact caused by the buffer. Next to that one is the positive control, and that shows a clear band confirming that the PCR worked as expected and amplified the targeted E. coli gene. Next to that one, we have food sample 1 and then food sample 2, and both of these lanes show visible bands of similar size, suggesting that the targeted E. coli gene was successfully amplified, indicating the presence of E. coli DNA. And then the last one on the right is the DNA ladder. This serves as a reference for fragment size. The bright bands correspond to known DNA sizes, allowing us to estimate the size of the amplified products in the other lanes. Conclusion: the results of this lab confirm the successful detection of E. coli DNA in the tested food samples using PCR. The DNA ladder provided accurate size reference points for estimating the amplified products, while the visible bands in the lanes FS1 and FS2, which were our food samples, demonstrated the targeted E. coli gene was present. This suggested potential contamination in those samples. The presence of a clear band in the positive control confirmed that the PCR process worked correctly and supported the reliability of the results. Although there was a faint band in the negative control, this was because of the use of a TAE buffer, which can produce nonspecific fluorescence from trace nucleic acids or impurities and doesn't indicate PCR contamination. These results show the effectiveness of PCR in identifying E. coli contamination in food samples, highlighting the importance of accurate molecular diagnostics and food safety monitoring.